Regarding applications of this method of siRNA usage within the research, it has wide applications. We can use this method to study the pathways, different communication pathways within the cells. When we culture the cells and we want to see the effect of any drug on those cells, like for example, if we are working with the cancer cells and we want to see that which kind of drug has what kind of effect, how quickly it controls or interferes with the cell cycle, and then subsequently we want to see that which of the gene plays important role in all these signaling pathways, the siRNA method is quite useful to perform this kind of studies. Then if we are working with the human disease samples, or let's say human disease models, we again want to see that which pathways crosstalk with each other, and if any of the protein is blocked, how efficient or how effective that blockage could be during that disease conditions, and ultimately we can control or we can improve the quality of a patient's life. In vivo conditions में if we use siRNA, and that siRNA is used in those animals which we intentionally developed having a specific gene, again we are going to interfere some genes, some proteins at the translation level, and we are going to study, we will see that what kind of effect this interference, this silencing can have on the diseased animal, and ultimately what kind of therapeutic approaches we can use, which protein we should target and suppress to stop or to interfere with the diseased mechanism. When we talk about usage of siRNA, we should have some methods to introduce the siRNA into the cells. For that purpose, various methods can be used, and one has to decide, keeping in view the target cells, the experimental conditions, and sometimes the other factors during the experimental setup. For example, we can use cationic lipid or polymer based transfection method, electroporation, or some chemical modification methods can be used that helps the siRNA to enter into the host cell. This table shows the technique, the delivery mode, advantages and disadvantages of that particular method during siRNA gene silencing. Transfection, electroporation, viral mediated delivery, or modified siRNA. Every method has its own advantages, has its own disadvantages. Transfection basically deals with the cationic, liposomic, or polymer based delivery method. Electroporation deals with the electrical pulse method for delivery. Viral mediated delivery needs some viral vectors. Keeping in view the cell type, resource availability, and the minimum hazard to the researcher, one has to decide which method should be used for entry or for delivery of the siRNA into the cells. Most commonly used method for transfection for entry of siRNA into the host cell are the cationic, liposomic method or polymer based methods, or sometimes we also use electroporation, the electric pulse generates the pores into the plasma membrane. So with this kind of information, siRNA can be used to silence the gene expression, to observe the role of that particular protein in cellular activity. That can be at functional level, that can be at phenotypic level, that can be at signaling level, and so on.